Hello, everybody. Thank you for joining this Elveflow webinar on microfluidic three-dimensional cell culture. My name is Lisa Muiznieks, and I'm a biofluidics application specialist at Elveflow. My colleague, Barbara Corelli, is a sales engineer for biology applications, and she will be joining us for the Q&A session at the end of the presentation. I ask that you please turn off your microphone and camera during the presentation. If you have any questions, you can write them in the chat box at any time, and we will answer them at the end. A few words about Elveflow before we start. Elveflow is a startup company whose goal is to develop state-of-the-art instruments for researchers. We are researchers ourselves, and our mission is to make microfluidics more accessible to specialist and non-specialist end users so that researchers from all disciplines that benefit from microfluidic flow control can advance the state-of-the-art in their fields. In this presentation, we will first introduce the use of microfluidics and flow for dynamic cell culture, with a focus on the advantages of culturing cells in a 3D environment, and describe on-chip strategies for creating 3D cultures. Then we will demonstrate how to set up and control flow to achieve stable molecular gradients, and finish by describing some research applications of microfluidic 3D culture before our Q&A session. Firstly, why use microfluidics for cell culture? While 2D culture platforms have been widely used for decades, they are greatly limited in capturing the physiological relevance of living organisms. Tissues are comprised of a dense and crowded milieu of cells, matrix fibers, dynamic fluids, and fluidic channels. Microfluidics is perfectly suited to adding some of these topological, mechanical, and fluidic stimuli to cells in culture to increase the physiological relevance of in vitro tissue models. For example, microfluidic perfusion mimics physiological flows such as blood flow and flow through porous tissue networks, known as interstitial flow. Flow provides nutrient exchange and removal of cell waste products. It allows the generation of sustained molecular gradients and adds important mechanical stimuli to cells. Not least, the smaller tissue scale of microfluidic platforms is also ideal for increasing the dimensionality of the culture environment from 2D to 3D, providing an extra dimension of relevance for the cell microenvironment. So as the term suggests, perhaps the most obvious difference between cell culture in 2 and 3D is that in 2D, cells adhere, spread, and make connections that are limited to a single flat XY plane. The 2D plane also exerts an unnatural polarity to cells. The culture surface is typically hard plastic, and the medium is homogeneously distributed in the flask, so molecular gradients are missing across the cells. 3D culture environments provide more relevant topological and mechanical cues, including a matrix to support the cells and to provide more relevant stiffness, porosity, confinement, hindrance, and ability to make cell matrix interactions. The 3D porous networks also enable the formation of molecular gradients. 3D culture environments provide the ability to support more than one cell type for more complex co-cultures. Growth in 3 dimensions opens the door to study the formation of 3D structures, for example, vessels that may have perfusible lumens. The 3D matrix itself provides an excellent model of interstitial flow, which is the flow of fluid through tightly woven tissue networks outside of blood vessels. In the lab, matrix pore size can be tuned. The generation of stable molecular gradients across the porous matrix enables chemotaxis assays to study cell migration in response to a stimulus. Microfluidic culture platforms are also suitable for generating 3D cell spheroids, or clusters of cells, for high-throughput drug screening. The spherical shape itself provides a more relevant model of drug distribution and penetration into the core of a tumor, and the control of spheroid size enables homogeneous replicates for comparing the efficacy and toxicity of different drug concentrations. There are many ways to produce a 3D cell culture environment. The main considerations are what matrix material to use, and if you start with the matrix or with the cells. The following description is by no means exhaustive, but we focus on setups that use and can benefit from microfluidics to make 3D culture models. 3D cultures commonly take the form of cells embedded in a polymer-based scaffold, which is a design derived from the native extracellular matrix of tissues. Commonly, polymer chains are cross-linked in the presence of water into a 3D hydrogel. Now, a wide range of polymers can be used, both natural or synthetic. Some common polymers include polyethylene glycol and PEG copolymers, as well as alginate and collagen. Pore size and mechanical stiffness can be tuned via crosslink density. Dedicated channels can be added to flow liquid through the middle of the gel, mimicking tissue vascularization. Open channels can be made using a needle or an embedded thread that is removed once the gel is set. Cells can be polymerized in the gel itself, as well as being seeded in the open channels. Alternatively, cells can be encapsulated into 3D droplets made from collagen or alginate, for example. This offers physical protection of cells and controlled release of contents. Droplets can be made using a microfluidic flow-focusing chip, using, for example, an aqueous alginate solution containing the cells and a cross-flow of oil to pinch off highly monodispersed droplets containing a single or several cells. Once the droplets polymerize into a hydrogel, they can be implanted into the body to aid wound repair, where encapsulated cells do not migrate as quickly away from the wound site, or as clinical cell therapies, such as treatment of Parkinson's disease or diabetes, where specialized cells can produce and release a therapeutic agent. Cell spheroids offer another type of 3D culture environment, whereby clusters of cells clump together to form a spherical volume. Cells in the spheroid secrete their own matrix and so don't require any scaffold to be added first. There are many ways to make cell spheroids, from off-chip, batch, and well plate methods to on-chip microfluidic methods. Using microfluidics, cells can be flowed through channels and left to deposit into microwells. Once formed, the spheroids can be used for high-throughput drug screens by flowing drug gradients across the chip. Spheroids made on chip have controlled and reproducible size, ideal for comparative studies of drug penetrance and efficacy. But the main focus of today's webinar will be the 3D hydrogels made on chip. Let's now look at some microfluidic chip designs that enable control of molecular gradients over cells in 3D culture. So a typical chip for working with hydrogels has multiple separate channels that run side by side that are interconnected lengthways. This might be by a fixed line of pillars with small spaces between them, or by a series of thin, flat microgrooves, here shown in black, connecting the central green channel with the red channels on either side. Now, since all these channels in these chips are interconnected, if you were to add liquid to one channel of an empty chip, the liquid would immediately fill the whole chip. Instead, the gel layer should be loaded first and polymerized. The gel acts as a semi-permeable barrier to the channels on either side and is a good model of interstitial flow. Here, the gel is represented as green balls and the flows are in pink. The unpolymerized gel is viscous enough to only fill one channel and not slip through the pillars. Adjacent channels can even be separated by a removable barrier, such as threads that can be pulled out after the gel has set. Now, in general, this type of chip design is flexible enough that you can put your cells in whichever compartment you prefer. You can load cells with the gel phase in a central channel, or create a 2D monolayer in a fluidic side channel, or do both. The composition of liquids flowing in the side channels dictates the concentration gradient of molecules across the chip. But how is the movement of fluid controlled, especially of multiple interconnected flows in a single chip? In the body, the heart is responsible for pumping blood. In the lab, flow is most commonly controlled using syringe or peristaltic pumps, or else using gravitational systems like a rocking platform. However, for increased stability and better response time, you can use a pressure-driven flow controller like the OB1 from Elveflow. Pressure-driven flow works by controlling the precise amount of air pressure on the surface of a liquid in a reservoir. With increasing pressure, the liquid is pushed through the outlet tube. The greater the pressure, the faster the flow. The pressure controller is hooked up to liquids in reservoirs and connected to the microfluidic chip with tubing. A bubble trap can be connected to remove bubbles from the liquid, and a flow sensor with a feedback loop to control the flow rate of fluid through the chip via a sensor mode as an alternative to controlling via pressure. The ESI software allows you to add different flow profiles through the chip, from steady to pulsatile to more complex customized rhythms like a heartbeat. And to perfuse a second or more channels simultaneously, you duplicate this configuration, shown here using a blue liquid. And indeed, Elveflow is a partner in the project Organoids, funded in France through the ANR under the support of the EU Joint Programme for Neurodegenerative Research, JPND, to study therapies for neuronal injury using a 3D multicellular organoid model. Elveflow is adding precise microfluidic flow control to enable dynamic perfusion of the neuronal model with drugs. So now we bring you into the lab to demonstrate the connection of the microfluidic instruments to the 3D culture chip. We start with the OB1 pressure-driven flow controller on the bench. The OB1 is connected to a compressed air source. Next to the OB1, we have a heated bath to keep liquids at 37 degrees and a CO2 incubator for the cells on chip. So first, we connect pneumatic tubing from the pressure-driven flow controller to a liquid reservoir. Then we connect the PTFE tubing for liquid from the reservoir to the bubble trap, which traps and removes any small air bubbles. Then we connect the bubble trap to the flow sensor, which measures and controls the flow rate via a feedback cable. Then we connect a piece of resistance tubing, which offers resistance to flow due to its much smaller inner diameter, enabling better control over flow stability. And then we connect to the chip. Now this liquid will be perfused through one of the side channels of the chip. To add flow to the other side channel, we duplicate the setup to connect a second liquid reservoir, bubble trap, flow sensor, piece of resistance tubing, and finally to the second side channel of the chip. And our tubing is cut long enough to be able to move the chip into a CO2 incubator. This control of stable pulseless pressure and flow rate feedback is ideal for generating and maintaining stable molecular gradient across the gel. Here we show a microfluidic chip with two flow channels, distinguished by yellow and blue liquids, separated by a central porous hydrogel that has been polymerized with polystyrene beads to represent cells. With stable flow control, we show formation of a green gradient from the yellow and blue liquids. Now, in the last few minutes, I'll describe some state-of-the-art research applications of 3D cell culture on a microfluidic chip. First, vessel formation in a hydrogel, including in the absence or in the presence of tumor cells. Then, migration of immune cells towards cancer cells embedded in a 3D gel. And last, a model of breast cancer metastasis into bone. The first study used 3D culture to investigate angiogenesis, or the formation of new vessels from existing vessels. This work is from the lab of Christopher Chen at the University of Pennsylvania. The researchers created a chip with two parallel channels, 1 millimeter apart, running through a collagen hydrogel. Endothelial cells were added to one channel and cultured to form a confluent layer, mimicking a vessel wall, shown schematically in green here, and stained in green in this image. Medium was flowed through this vascular channel. Different angiogenic factors were flowed through the other channel to generate a molecular gradient across the collagen matrix. Certain molecular cocktails promoted the formation of sprouts from the parent vessel all the way to the source channel that developed perfusible lumens, shown by the movement of fluorescent beads through the new vessels without them leaking into the interstitial space. This model was used to capture morphogenic steps of angiogenesis, and the 3D culture environment provided key molecular gradients to study chemotaxis. Vessel formation was also studied in this paper by the Hughes Lab from UC Irvine, California. Vascular genesis is the formation of new vessels from progenitor cells. Here, the central channel took the shape of 3 connected diamonds and was filled with a fibrinogen hydrogel containing endothelial cells, in red here, and stromal cells. Side channels were perfused with medium and/or growth factors. By day 7, a tight vascular network was formed, and flow across the gel from one side channel to the other switched from being interstitial to intraluminal, shown by the flow of Fitzy-Dextran here in gray. Now, because tumors rely on vessel formation to support their high metabolic activity, next, the researchers added tumor cells to their 3D vascularization model. The cancer cells, in this case from colorectal cancer, formed tumor-like spheroids, shown in green in the gel, supported by vessels, shown in red. Perfusion of the vessels with a model anti-cancer drug cocktail reduced the tumor growth compared to the control. And 4 days after removal of the drug, the tumor growth was still suppressed. Microfluidic 3D culture was key to generating this model of microtumor and vessel formation due to the ability to apply molecular gradients to promote cell migration and organization, while the tumor spheroids provided a more physiologically relevant volume for drug studies. The next example used 3D cell culture to model the tightly connected crosstalk between cancer and immune cells to evaluate the efficacy of immunotherapies. The study is the work of Parletto and colleagues from Rome, Italy. Here, the researchers used a chip with 5 channels. The 2 outside channels in purple contain medium. The 2 red channels were filled with a collagen gel with or without cancer cells. And non-adherent dendritic cells were added to the immune chamber in the center in green. The immune chamber is connected to the tumor chambers on either side by microgrooves that mimic the physical constraints of cell migration, such as extravasation. The researchers tracked the migration of dendritic cells through the grooves into the gel, preferentially towards the tumor cells. They identified that the immune cells modified their trajectories in 3 dimensions in order to reach, then interact with, and phagocytose the tumor cells. Here, a dendritic cell in green is engulfing a tumor cell. The 3-dimensionality of this co-culture model was essential to incorporate the key physical constraints and biochemical gradients that play a major role in the immune response to tumors. And the microfluidic setup enabled the addition of non-adherent cells. The final example used a 3D microfluidic culture to study the metastasis of breast cancer to bone and the mechanoregulatory role of bone cells in this process. This is a study from the lab of Lidan Yu from the University of Toronto in Canada. The researchers developed a 3D vascular channel of collagen lined on the inside with a confluent layer of endothelial cells and an adjacent fluidic channel cultured with a layer of osteocyte-like bone cells. The 2 channels were connected via triangular pillars, enabling the movement of components and the generation of chemical gradients across them. Breast cancer cells, here shown in green, were added to the vessel lumen. The vascular channel is also shown here in these micrographs, with the endothelial cells in red and the cancer cells in green. Next, an oscillatory fluid flow, or OFF, was applied to the bone cells to stimulate the osteoclasts, mimicking exercise and natural bone homeostasis and remodeling. The mechanical stimulus induced an intracellular calcium response by the osteoclasts, and it correlated with substantial reduction in cancer cell extravasation, an intracellular calcium both in the number of cells that moved and the distance that they moved, compared to no mechanical stimulation. Again, the microfluidic 3D culture environment enabled the formation of chemical gradients, this time induced by a mechanical stimulus, to study disease progression and potential therapies. So these examples illustrate some exciting advances in cell and tissue models, realized in part due to the added dimensionality brought by 3D microfluidic cultures. So in summary, microfluidics is ideal to add flow to cells in 3D culture. High-level flow control using the OB1 pressure-driven flow controller and flow sensors with precision feedback enables perfusion of interconnected channels for generating molecular gradients and models of interstitial flow. Perfusion of 3D models enables the study of chemotactic and mechanosensitive cell migration for studies of metastasis, immune response, vascularization, as well as facilitating the addition of non-adherent cells and pulsatile flows. So if you'd like to know more, we invite you to check out our range of application notes using microfluidics for cell biology on our website at elveflow.com. We also have detailed protocols, including how to produce alginate particles or to recirculate medium over cells and culture. Or feel free to contact us at contact@elveflow.com. We also point you to our online microfluidic calculator tool to help you calculate flow rates, pressures, and suitable resistance to use with your specific setup. So with this, we thank you for listening. And now, together with Barbara Corelli, we'll open the floor to you and invite you to ask any questions you may have via the chat box. And we will try to get through as many as we can. Thank you.
So hello, everybody. So let's see the first questions. So Amira, hello, thanks for this great presentation. I was wondering, how do you prevent a clogging to occur in the device?
So the whole idea here, I think, thanks Mira for your question, is that the gel itself is entered into the chip through dedicated ports. So the gel itself has an inlet and an outlet that you enter the gel into and polymerize it in the chip. And it's the side channels on either side which you flow the liquid through. So in this way, the standard procedures, for example, filtering your solution will help you with stopping any clogging.
Yeah. So another question from Alexandra. Thank you for an interesting talk. How much does the setup shown on the film cost? So to answer this question, I invite you, Alexandra, to send us an email at contact@elveflow.com. And actually we can discuss that also because our system is highly customizable. So we can really check first what are exactly your requirements and what is your application, and then we can see what will be the final cost. Alexandra, thanks a lot for your interesting presentation. Thank you. Could you please explain a bit more how we could use 3D in Parkinson's disease?
Well, as far as I understand, certain diseases require cell therapies, for example, to release therapeutic molecules. So if you had cells that produce these therapeutic agents and you encapsulated them inside a particle that was, say, made of alginate or collagen or something to protect the cells, you could then use these particles loaded with cells that would produce therapeutic agent and implant them into the body. And I think this is a powerful tool and quite new, I think. Yeah.
We have a question from Nehal Desai. Hello, thanks a lot for the insights into the microfluidics. May I know how this field can help in COVID-19 or in the vaccine production? Basically, as you might know, some of the vaccines that have been produced, especially the RNA-based ones, are based on the use of lipid nanoparticles. So this is something that you can achieve with microfluidics. So producing, for instance, lipid nanoparticles and deliver then the RNA vaccine through that. So this is a tool that can be used definitely for COVID vaccines. I go back to the question of Marina Rana. Thanks for the amazing talk. I have a very specific question about my system. So I'm using lipid oil solution in the chips and wanted to reuse it. What do you think is the better way, to wash the chamber without rupturing it? To be honest with you, so the standard like cleaning procedure for chip are mainly using some disinfectant, water, washing with ethanol and air dry. I don't know if you Lisa have a specific tip for that in particular.
No, that's how I would wash the chip for cell culture. So I'm not quite sure, again, your protocol for the lipid oil solution, if you can remove it from the chip, wash the chip and re-put it back in. I'm not sure.
Yeah. Maybe, maybe trying with just first disinfectant and then water and ethanol, and let's see. Another question: how did you design the chip?
So yeah, you can do different things. There's different strategies. You can design your own chip and fabricate it yourself if you have microfabrication facilities where you are. You can, for example, pouring with PDMS, or you can buy commercial chips if you prefer that already have the design already, so you don't have to worry about designing anything specific, and you can use something that's around. So it depends on your application. I think there are some chips around with 3 side-by-side channels separated by pillars. So you can already use things that are off the shelf.
Yeah. Another question from Amira: do you provide a specific microfluidic kit to perform a 3D cell culture? Thanks a lot for the question. So indeed, Elveflow, we are providing you with everything to control the flow around the microfluidic chip. So we can definitely provide you with the kit for handling the liquid part, and give you tips and suggestions for the chip part, for some commercial options that we have tested internally. So you can already have a look at our Organon chip pack page on the website, and I invite you to send us an email at contact@elveflow.com to know more. Samara, do you have a multi-channel chips? As said, Elveflow provides everything for controlling the flow before your chip. We can give you really good tips on which chip you can use for your application, commercial options, or even custom ones, multi-channel too. Another question from Abdi Nandan. Thanks. One more question. Can you suggest microfluidic solutions for live and dead cell separation?
That's a tough question. I mean, it's a tough problem. I mean, thanks for your question. No, I'm not sure. Tracking live and dead cells, obviously using dyes is one thing. Staining with trypan blue or propydium iodide or something, and actually counting them to know which cells are alive and which cells are dead is one thing, but separating them, no. Not sure.
No.
I think that's a hard topic. Sorry.
Definitely. Another question from Mitra Akbari. Hello, can you please explain the difference of 2D and 3D chips?
All right, so there's not necessarily much difference between them. You can have, 2D chips often have just one channel, for example, because the liquid is free to move through the channel. The cells are loaded or seeded first, and then the chip is held static for a few hours, or 6 to 24 hours, depending on your cell type, for the cells to adhere. And then afterwards, you flow fluid through one channel, for example. The 3D chip, the real advantage is having separated channels that are interconnected with, say, these pillars, so that you can add gel in the center, and then that acts as a barrier between the flow on either side. So I think that would be the main difference between those chips.
Yeah, and also I can add just one point here. So you can have also some 3D chips where you can exactly have 2 flows and the membrane in between, where you can grow a type of cell on one side and a type of cell on the other side. So you can really create a co-culture, so multi-layers, let's say, sample, I would say. Another question from Flora Magno. Thanks for the presentation. I'm new in this field and it was very clarifying. Thanks a lot. So is it possible to create vascularized brain organoids with microfluidics?
So I think the idea would, organoids are quite specific. I think the idea is, yes, if you have the cells, you can try. It would obviously require some optimization and whatnot. But if you have the cell type that you wish to form into an organoid, it will be possible using microfluidics. Yeah.
Yeah, in general, I would say as a general comment that when you get your chip, as soon as you have your specific cells that you want to grow into it, then you can definitely recreate any kind of organ, let's say, and applying then a flow to mimic all the dynamics and interstitial flows or mimic endothelial cells and vascularization then accordingly. Clarissa Bescher, thanks a lot for the question. Thanks for the talk. Do you know for how long can you guarantee a stable pressure control with the OB1 controller? Is there a time limit in days? So actually, you can really perform longer-term experiments with the OB1, and I let Lisa tell for how long you run experiments with the OB1.
Well, I agree with Barbara that the problem or the limitation is not necessarily the OB1. That's very stable for as long as your system is stable. So long as your gel doesn't break, or so long as your cells don't overgrow, for example, or
and if we can quantify, no I would say that at least days or a week,
for example. Yeah.
Another question from Abhinandan. Can you share us the presentation and link to the video after the session? Yeah, definitely we can do that, and everything will be available soon on our YouTube channel actually as well. Flora Magno, thank you. Thank you to you as well. Main Rana, another question. Without the flow control device, how can I decide on the pressure for different inlets? I mean, I have 3 inlets, 2 for the lipid oil and 1 central for the aqueous phase. How do I decide on the pressures? Okay, so basically this is really the feature of our instrument. So with our OB1 controller, you can have 3 independent pressure channels that will be able to control independently your 3 fluids in your 3 inlets. And you can on the software set up the pressures as you wish independently for the 3 inlets.
Yeah, you're right. Without flow control device, it's more difficult. So
yeah, well, of course, but it's true that with the OB1 you will be controlling the pressures, and with the flow sensor the flow control. Another question from Abhinandan. Does syringe pump vibrations affect flow inside microfluidic chip, pulsating flow? And do you provide any solution to overcome such issues faced using syringe pumps? Thanks a lot for the questions. So actually, you're right. Actually, using syringe pumps, you may have this issue. That's why we invite you to have a look at our systems that are pressure-driven flow controllers that overcome this issue, because they are able to provide a really stable and smooth flow. Based on the fact that we are relying on moving fluids through pressures and not through mechanical actions like using syringe pumps. Okay, so if there are no more questions, we thank you a lot for participating to this webinar, and don't hesitate to contact us and send rating to this webinar. We will be happy to help you and propose you some options for 3D cell culture experiments in dynamic conditions. Thanks a lot.
Thank you, everybody.
Thank you, everybody. Have a good one.